Okay, good morning, everybody. Took me a second to get it all switched to the right thing. All right. So there truly, I feel like, is a plethora of information when it comes to respiratory tract infections. And really, not that other body sites aren't important, but when I think about a lot of the infectious disease, respiratory tract infections and digestive system infections, both of those body sites are huge when we think about infectious disease. There are all sorts of culprits and numerous things that I feel like we could go into great detail with. So with the respiratory tract, again, a tremendous amount of potentials. And we're going to divide it into a couple of areas, some of which we might expect to see the potential for more organisms to say, I want to live there, and others that really ought to be more sterile. So upper respiratory tract, we think of as a suitable habitat for many microbes. And as we move into the lungs, the lower respiratory tract, typically we think normal flora is something that's not really observed there. And when we see something, that's typically our culprit for whatever respective situation we're dealing with. So, oh, so nasty. All sorts of aerosols as we see sneezing or coughing, just simply talking, which I know is why everyone's set so far away, right? So the potential for particles to be spewing out from someone, and with that carrying all sorts of different microbes, is truly a potential. So cover your coughs and sneezes, prevent that stuff. So quick anatomy. I'm not going to go through here and spout off all of this stuff. This first image shows the upper tract. Like I said, we're going to divide it into upper and lower. And the upper tract has the potential for numerous things to colonize. And a lot of that we think about good situations to help to keep us healthy. There are also some potentials where we may think about some of these things maybe living in the upper tract that sets the stage for potential issues if they can get down into the lower respiratory tract. Streptococcus pneumoniae is something that we might see in the upper respiratory tract, and then the potential for it to cause pneumonia if it makes its way into the lungs sets the stage for some serious issues. So, and handfuls of other things. We also have the ear, and we can see it's also connected into the upper respiratory tract, reaching into the nasopharynx through the eustachian tube. So we talked last time about some outer ear infections. Now we want to think more details about some potentials for other stuff, and it can be migrating up through the nasopharynx into the eustachian tube, causing middle ear infections, or inner ear for that matter. Just depends on the situation and who's participating. As we move down into the lungs, this is the lower tract, and as I said, typically thinking of a more sterile habitat, not that we can't see things living there, but typically stuff that shouldn't be, all right? So there is multiple potentials to try to keep things in check with mucus secretions and also ciliated cells that help to kind of push things up and out of there and prevent them from making their way in. When we talked immunity stuff within the lungs, there are macrophages, these alveolar macrophages residing in these tissues to help recognize some that maybe slip through the mucus traps and the cilia that are trying to whip things up and out of there. The macrophages are present with the hopes that they can combat them before any sort of infection becomes established. And that's what we're looking at here with these ciliated cells and the goblet cells that are secreting the mucus. Here's the lumen of the trachea. And so this providing with that innate type of immune response, providing a really first line of defense against some potential things we may be coming in contact with. So we showed in the previous chapter some details with Streptococcus pyogenes when we looked at some skin infections and flesh-eating stuff. And I mentioned last time that really when I think about Streptococcus pyogenes, I think of it more as a respiratory situation, sore throat or strep throat. Super common culprit is Streptococcus pyogenes. There are numerous other things that can cause pharyngitis, but it's definitely one that's a key player and something that warrants antibiotics and the hopes of preventing issues. We've talked about like immune complex disorders, and a good chunk of those things, we think about some of the immune complexes formed against pyogenes. And so that sets the stage definitely to warrant an effort to try to combat these as they take hold. With that said, there are also some opportunities for individuals to harbor some of the Streptococcus pyogenes in their throats as normal flora, low levels, and they're kept in check by other organisms, preventing them from truly proliferating and causing issues. And we would recognize those individuals as carriers. They don't exhibit any symptoms of strep throat, but they have potentials to maybe transmit it to someone else. We could flip it back to that guy sneezing and spewing out all sorts of particles. Maybe something like that sets the stage for these to be spread about relatively easily. So I do like this image of the nice chain appearance of our Streptococcus pyogenes. And there's a couple of bits in here where we can see them in the process of replicating. And I think that's pretty cool. You can see a few of them right there as well. So here we could see in the back of the throat an individual with Streptococcus pyogenes. These red little splotchy bits are called petechiae. Streptococcus pyogenes is a bacterium that we describe as beta hemolytic. I've mentioned that stuff before. A bacterium that's beta hemolytic has the potentials, if blood is present, to destroy it, to lyse those cells and basically suck up the juices from those red blood cells. It likes that. And this is really kind of evidence of that type of reaction, the hemolysis reactions as it's getting in there and trying to get access to some of those nutrients. There's also potential, though we don't see it really in this image, but maybe back here by the kind of tonsil region, some whitish growth or pus back there. That may also be relatively indicative of something like that. Bacterial, for sure, we start thinking, all right, whether or not truly strep, that's to be determined. Rapid strep tests, they go back and swab. Here's the uvula. You want to be close, but you don't want to touch that. You want to be back here by the tonsils and trying to get some of this area not touching the tongue when we do this, and hopefully lending to the potential to see some of these. We would inoculate it onto a blood agar plate if we were swabbing it for growth. The rapid test on that swab gets added to some reagents, and basically an immune response showing the presence of these antigens, the Streptococcus antigens, will give you a positive marker on the little filter paper as it moves across it. So typically, strep throat stays localized within your throat, and we don't think of it causing other issues. But there are definitely potentials for numerous complications. I already mentioned some of those immune complexes. Scarlet fever is a skin rash that we can see as the result of streptococcus infection. And there are some variations with this to see it presenting. And I believe, I could be wrong, but I'm pretty sure the ability to progress to this relies on the bacterium being co-infected, or not co-infected, but infected with a virus that stimulates it to manifest in this fashion. Pretty sure that's the situation with it. So here we could see, shifting from that to the ear, a normal ear. As we are looking in here, we could see the eardrum or tympanic membrane and some of the bones behind there, the malleus and incus, the stapes, someplace we're not seeing that, but sort of, right? And here's an image where it's an ear infection with a torn tympanic membrane. I recall just seeing this with the little bones inside of the ear. There was a person that worked in the anatomy labs. This was years back, and she worked with the donors, as you know, preparing them for A&P labs, getting stuff ready, doing different dissections and things like that. And she was super excited one day to be able to get all three of these removed, and they were all intact, super small. But she's like, I got them, right? And I was just like, okay, right? I geek out on stuff like this, the grossness, right? She's geeking out on the, you know, ability to dissect and remove, and I'm like, it's the same but different, right? So I always, as I see this, it always reminds me of her, and I like not really seeing the bones. It always reminds me, she extracted those things. So diphtheria, Corynebacterium diphtheriae is the culprit for this. We'll see a table with details. I'm just quickly trying to give you some of these images, and then we'll hit some of the table stuff and think more about these. But diphtheria is a bacterium that can create this pseudomembrane at the back of the throat. And here we can see this. It's all sorts of stuff. Dead cells, pus, fibrin, red cells, and the bacteria as well. That all are kind of in this mass of kind of nastiness. And that's something that we think about as a kind of a characteristic with diphtheria. And we'll talk about some of the details with that as we progress along. As we move in and think stuff like lower tract infections, we think often lots of types of pneumonia. There are numerous culprits that we can think about that stimulate these situations. So we see lesions, opaque areas within the lungs where we've got fluid, mucus, all sorts of bacteria that are able to be visualized with a radiograph. One of, just like thinking, you know, pyogenes and strep throat as a typical player, oftentimes when I think about pneumonia, I think about Streptococcus pneumoniae as one of the key players, but there are truly a number of organisms that can cause complications. So Streptococcus pneumoniae, a cousin of pyogenes. It's one, if we were to look at it under the microscope or an electron micrograph, but under the microscope, instead of big long chains of positive cocci, it's two together, diplococci. And you can search and search, but it's almost always just groups of twos all over the place, right? Never big long chains. And that's something that helps to maybe allow you to differentiate just based off of some staining. There are other tests that we might do if we were truly trying to clue into differences between pyogenes and pneumoniae, and some of it is some antibiotic susceptibility testing, where we can separate the two based off of their reactions with some of that. But we also think more about some of these situations. If we're thinking pneumonia-like symptoms, we would be thinking more likely this. If it was a simple throat swab, the potential to find both pyogenes and pneumoniae could truly be present in those situations, right? And then you'd need maybe to do some further identification if we wanted to see some of that. So Haemophilus is a bacteria that can cause numerous things. Haemophilus influenzae, as we see here, growing on a chocolate agar plate. So this is a type of media that it's not, as the name implies, it's not truly chocolate. It's red cells that have been autoclaved or cooked, causing them to basically bust open. So it's cooked blood, basically, and it makes it have this dark brown appearance that looks chocolate-like, all right? I bet you it doesn't taste like chocolate, all right? So there are numerous bacteria that like feeding on destroyed red blood cells. They like the nutrients that they can get from that, but they can't destroy those red cells on their own, like the beta hemolytic streptococcus or others that are also beta hemolytic. Haemophilus wants to grow on that stuff, but it needs it to be destroyed for it to grow, all right? And so the chocolate agar gives it those nutrients. If you tried putting it on a blood agar plate by itself, it wouldn't grow because it can't get all of the nutrients that it truly needs. There are some potentials, and maybe you're in a lab where you're suspecting something like this, but you happen to not have chocolate plates on hand. These are typically things you purchase, not make in the lab. If you have blood agar plates, you can take a bacterium that's beta hemolytic. Usually we use Staphylococcus aureus. It's a good beta hemolytic bacterium. You can take a swath or a strip of Staph aureus down the middle of the plate, and Haemophilus can be kind of zigzagged or fishtailed through that swath of aureus. And where the Staph aureus lyses those red cells and creates the clear zone, it's also releasing those nutrients that the Haemophilus can use to grow. And instead of it being these beautiful, big, shiny colonies, they're little teeny pinpoint colonies on those lysed area in the blood agar plate, and we call that a satellite phenomenon as the Haemophilus is growing around the Staph aureus. In this image, with these shiny colonies, we can see the potential that they have for the sugar layer, the capsule layer, that gives them a potential to avoid immune detection. And these are good ones when we think about stuff like, respiratory tract infections. It's also, we saw Haemophilus as a player with conjunctivitis last time. Mycoplasma pneumoniae is a bacterium that's odd. I believe I have mentioned this one before, as an organism that doesn't have a cell wall. It's something that we describe as being pleomorphic, so the ability to kind of take on different shapes. Here we can see mycoplasma as it's trying to dock or attach to epithelial cells in the trachea of a hamster. So it's got some ability to link up to those receptors, and then as it attaches, it establishes the ability to start causing some disease in whatever, all right? So these are things that, without a cell wall. They don't gram stain. We would do some negative staining to try to visualize them. They are very small, considerably smaller than, like thinking stuff like E. coli that's really pretty little when you're looking at it under the microscope. Mycoplasma is even potentially smaller, and part of that's the lack of cell wall that makes it difficult to visualize. As you see this ruler bar over here, 500 nanometers, half a micron. That's about how big that is, right? Not so big, all right? As far as the pneumonia that it causes in people, it's something that's maybe a little more mild. We oftentimes call it walking pneumonia because it is something that isn't so debilitating that she just flat out can't even get out of bed, all right? You're able to kind of function. You still feel ill, but not so bad like we might think of with numerous other pneumonias. We talked a little bit about tuberculosis before. These bacteria are acid fast, so they've got the mycolic acid that surrounds the cell that makes it really pretty impervious to numerous types of stains. Gram stains usually don't show any reaction. If that cell is maybe a little, if that culture, those cells are a little bit older than a nice fresh one, some of those cells might be in the process of starting to die. The mycolic acid might potentially start breaking down the littlest bit. And sometimes they pick up a gram stain. They do have a thick peptidoglycan layer and would appear with that mycolic acid layer kind of depleted, they would appear gram positive. But typically in a fresh culture or a new sputum specimen from an individual, you're not going to see a gram reaction. You're going to then question other things and maybe do acid-fast staining, and that shows the acid-fast bacilli that's indicative of tuberculosis. It is truly the only infectious or bacterial infection that we can recognize and identify based off of the acid-fast staining, and we start treating with antibiotics before we've ever truly cultivated it and gone through the antibiotic susceptibility, because it is such a dangerous pathogen. They do have numerous potentials for survival, forming tubercles within the lungs, within the alveoli, as we see in the image down below, while being walled off by some of our immune responses, forming this granulomatous reaction, encasing them in sort of a shell, but it's not something that is killing them. It's just kind of blocking them. They've got the potential to disperse, and this in turn sets the stage for lengthy antimicrobial therapies as the result of tubercle formations. They also can take up residence within the alveolar macrophages, those cells that should be destroying stuff. Tuberculosis gets in there and hangs out, replicates, and destroys those macrophages to go on and spread. All right, so a couple of kind of brilliant strategies that lends to success for this bacterium and issues with us. So here's a skin test. We talked a little of this with the last chunk of material for TB, the delayed hypersensitivity reaction, so cell-mediated type 4 response. So a couple of tables of bacterial culprits, numerous things, and like I mentioned last time, there are potentials to see some players in a handful of these chapters as we move through, and we definitely are seeing a little bit of that with some of these, and there's definitely some that are truly respiratory stuff. So the middle ear infection, or otis media, numerous players that can set the stage for this: Haemophilus influenzae, strep pneumo, moraxella, and some others. thinking respiratory, upper respiratory tract moving up through the, into the eustachian tube and into the middle ear. So bacteria that may be culprits in the upper tract that make their way in and cause problems. So earaches, effusion, fever, nausea, vomiting, and diarrhea, all potentials. And it also may be varied dependent upon the respective culprit. Oftentimes, these are a secondary type of infection where the bacteria that are thriving or growing, maybe not necessarily thriving, but are in the respiratory tract, make their way up into the inner ear or middle ear and cause issues. So there are some potentials with antimicrobial therapies. There are also some potential issues with some of these and maybe some biofilm that lends to problems with that, all right? And maybe lending to little kids with tubes in their ears to potentially prevent some of this stuff from being a recurrent problem. We saw the image with diphtheria, the cyanobacterium diphtheriae. There are all sorts of issues as we see the pseudomembrane. It can be within the lungs, lending to some serious complications and suffocation and death. So droplet transmission. There are some antimicrobials, but we also think about vaccination. The D in DTaP, and we will talk about our others as we move through here, but diphtheria, the tetanus, and the P for pertussis, all right, whooping cough. That's the three that we think about in this, and some potential deviations with that a little bit, dependent upon some of the vaccines, but typically it's a DTaP that drills you with all three. Legionella, Legionnaire's disease is Legionella pneumophilia. This is something that is relatively new on our radar of infectious diseases, something we would classify as an emerging pathogen. There are others like our tuberculosis that we would say is a re-emerging pathogen, something that we thought we had a pretty good handle on with antimicrobials to now see it flaring back up with some multi-drug resistant strains, something that we never truly got rid of, all right? There's likely been occurrences of some Legionnaire's disease before we recognized it in the 1970s, but that's when it really first popped up on our radar, and all sorts of issues with this pulmonary problems. It's something that we see transmitted through aerosols. We see it using water as a reservoir. We'll see an image in a minute with an amoeba. They like water and wet types of habitats, and the Legionella can survive inside of the amoeba, giving it a potential and using the amoeba as a reservoir. So misters, things like swamp coolers and air conditioning units, misters at grocery stores that spray produce, all of these can be prime breeding grounds to harbor some Legionnaire's disease, and inhalation of that may set the stage for problems. Oftentimes we think about maybe a weakened immune system setting you up to be more likely to become infected. So it's not like, coming in contact with it guarantees the infection, but it could, all right? So sometimes fatal infection and different antimicrobials with that. Pertussis, I mentioned just a second ago, the whooping cough, Bordetella pertussis, something that we're seeing kind of surging back up with some anti-vaccine issues lending to big problems with things like whooping cough, something that can be a fatal infection when we think about little kids, infants. This cough, whooping sound is a chronic cough that can last for months, and in turn, serious issues with that, as I said, potential fatal when we think about little kids. So respiratory droplets from that infected individual is what sets the stage for transmission of that. And vaccination, truly trying to keep these at bay. Our next one on here, Q fever, Q for query, because it was a big question mark. They didn't really know what the heck was causing these fevers, speculating some sort of bacterial origin, but didn't really have it pinpointed. So that's where it got its name. And then later discovered the bacteria Coxiella burnetii as the culprit for Q fever, which, as the name implies, you get a very high fever. There's coughing, pneumonia. There are some potential fatal endocarditis situations in some circumstances. So numerous things that can set the stage to come in contact with it. Aerosol transmission, so aerosols of urine, feces, milk, or amniotic fluid from livestock. Cattle, sheep, and goats all set the stage to potentially cause these problems. So strep throat we focused on as we started this, pharyngitis and scarlet fever, numerous symptoms with that, and relatively highly contagious when we think about it. Direct contact or droplet transmission sets the stage for problems with that. TB or tuberculosis, we saw some details with that. There's definitely issues with it. In the United States, we see cases of it, but truly when we think about other countries, it's definitely more of a concern. And antimicrobial resistance, big problems. People not complying with their drug schedules set the stage for more problems with that. Numerous issues when we think about it. There are vaccines as potentials. In the United States, we don't think about this truly as much of an option. Military and some other individuals potentially vaccinated against TB, but usually when we think about civilian population, not, right? If you are from some other country, might be something that is a common vaccine schedule, something that you'd likely be getting vaccinated against. Next, still, like I said, a couple of images of bacteria, or a couple of tables with the bacteria as we move through here. Chlamydial pneumonia, there are a handful of different culprits for this. Chlamydophila pneumoniae, and then cysticae, chlamydia trachomatis that we saw the eye issues before. This is a sexually transmitted infection. Here we see some others, and this potentially something thinking like birds as a potential. That's the infected bird. So bronchitis, and then potential for mild to severe respiratory distress. So droplets or aerosols from our pneumoniae, exposures to birds, or transit through the birth canal may set the stage for coming in contact with that and aspirating some of the bacteria to set the stage for it to cause these problems. So a couple of antimicrobials. Haemophilus, we saw it on that chocolate auger. Cough, fever, low body temperature. So fever is not something we think about here. Chills, chest pain, headache, and fatigue. So something that could be potentially contracted from an asymptomatic carrier, harboring it, like the potential for maybe some pyogenies as well. Klebsiella pneumoniae, this is one that, we've seen some bacteria producing capsules, and they're really shiny, really mucoid. Klebsiella is super good at producing some capsule layer. It produces very mucoid colonies. Oftentimes, we grow these plates up in the incubator and we flip them upside down. You guys have all been doing that, I'm sure. Lids go at the bottom, the bacteria is growing on that agar, and if you're streaking for isolation and it's flipped the other way around, condensation on the lid drips down on the plate and you get all sorts of problems. I have seen some Klebsiella pneumoniae growing on the plates, and as they're growing upside down, they produce such copious growth that the slime drips onto the lid as it's incubating upside down. That's pretty gross. And trying to open the lid and manipulate some of that with a loupe to pull some off and stain it up or whatever is kind of a snotty mess. It's truly kind of impressive to see that. So it causes lung necrosis, a current jelly sputum, which is ultimately the result of red cells within the sputum. And this oftentimes is fatal infection when we progress to this current jelly sort of sputum. You know there's some serious problems. Klebsiella is one that is a highly drug-resistant type of bacteria. And so careful antimicrobial screening to truly figure out the right drug of choice is necessary to ensure that we combat them. And oftentimes you're really racing against time to try to deal with this. So the walking pneumonia, mycoplasma, low fever, persistent cough. You don't feel super bad, like I said. So inhalation of droplets or aerosols from an infected individual. Strep pneumo is the pneumococcal pneumonia. There are all sorts of ill effects with this: productive cough, bloody sputum, fever and chills, respiratory distress. There are handfuls of vaccines, and the PCV 13 or 23, these are different valences, so different proteins. So it's kind of a cocktail sort of an approach that gives you the opportunity, because there are numerous sorts of strain variation. So this gives us the ability to try to mount a response against a handful of different fingerprints from those variants that gives us the hope that we've got a leg up against them. Another one that's pretty drug resistant and also pretty good at capsule formation like the Klebsiella is Pseudomonas pneumoniae. And we saw some issues with it before, and the green pigment and things like that. The real good capsule formation makes the mucus really thick and viscous. It can really cause some serious issues, especially in individuals with cystic fibrosis. Pseudomonas is a common culprit of colonizing, and it becomes really difficult to try to combat in those situations. And we see that right here with the potential. So nosocomial, definitely a situation with that, where we think about ventilators and the potential to spread that big issues. All right, so shifting to some viral stuff, and there are a handful that we want to think about. I will try to move through here quickly. We've got about 10 minutes. Here's an influenza virus, and there are all sorts of differences when we start thinking viruses, but oftentimes a few things that are kind of similar. Some spike proteins that we see here, these H and N proteins, not exactly the same, but different viruses with some similarities with the types of proteins that may in turn allow them to make recognition of respective types of host cells, whatever it is they're trying to infect. And as we're thinking about with respiratory secretions or respiratory infections, excuse me, these would like to make contact with cells within the respiratory tract and linking up and bringing them in. So in turn, as we think about these proteins on the surface of these viruses, we think about vaccine development, and it doesn't matter if we're talking influenza or other players. We think about trying to recognize some of these surface markers to mount immune responses against. And with numerous ones, the flu, as we've got here, we see all sorts of variation, different potentials to show every year with different strains of influenza, and potentially recombining some of this to create something that's even more different than the patient was infected with, that sets the stage for maybe something more serious with the respective combinations of proteins that are being shown. So I try to mount vaccines against that with the hopes that we're finding that proper combination, but sometimes it's not truly as effective as we would hope. Measles, another viral infection, or another infection. The first ones we were talking with vaccines were bacterial, but this, another infection that we think about vaccine to prevent in the first place. So there's two types of measles when we think about this. Regular measles and then German measles. I think that's the next image, right? So German measles, rubella are the German measles, and rubeola are the regular measles, right? So with this, technically we could back it up and be like, well, could we have put this in the previous chapter and talked about skin stuff? And definitely we see manifestations on the skin, right? So raised macular rash, we see it starting off on the face and then spreading to the extremities. You can see these Koplik spots in the mouth, another characteristic of measles. That's what we're looking at. And then here's the actual measles virus or virion, as we see it with the electron micrograph. German measles, the rash is more mild and not confluent, so they stay separate little bits versus these where they merge together to form bigger macular types of rash. And then here we can see some of the virions as they're budding out of host cells, so just being released, releasing all of these virions, which in turn will make contact with new cells and be brought in and go through the viral cycle. Here's some images of chicken pox. So this on the trunk of an individual, we can see these pustular lesions and the virion of it. It's a herpes virus 3. Varicella zoster is who we call it, but it is a type of herpes virus, herpes 3. Numerous herpes viruses and their respective manifestations varied as we will move through and look at them. So here we can see an individual with shingles. The lesions that we see with shingles are definitely different than the chicken pox that we see over in the image here, right? So it's a reactivation of that, and the virus, lots of virus can kind of go dormant, hang out on respective cells. Nerve cells typically are good places for some of these to just wait. And so shingles, when it flares up, it's typically showing up in a specific area where you've got one trunk of nerves that we see reactivated. There could be another area where the chickenpox has also gone dormant, but we don't see it as a systemic sort of thing. It's more of a patchy, maybe one side of your trunk or here, part of the face impacted, but the rest of the body likely free and clear. Just kind of depends. It also depends on some of the circumstances with this. Oftentimes we think about shingles popping up later in life 60s or so, when we might start thinking about some of that. And there are vaccines to prevent shingles, just as there are vaccines to prevent chickenpox, not the same vaccine. So here we've got a whole list, a handful of these that we've already looked at. So the varicella zoster or chickenpox, often thought of as a childhood illness, but with vaccination efforts, may be something not so much. My gut says a handful of you probably have been vaccinated and never saw it, right? Others maybe not, and maybe you did get some chickenpox. So kind of just depends. It is something that is highly contagious, right? And just simply coming in contact with an individual that's got it, and if you haven't been vaccinated or you haven't had it, you kind of expect that you're going to see it. So common colds, we didn't mention these, but there's a boatload of different common cold culprits: rhinoviruses, adenoviruses, and coronaviruses, and plethoras of each of these that set the stage for issues. Symptoms very similar, right? Most all of these very contagious as well. Influenza, 3 different types of influenza virus, also contagious. Fever, chills, and headaches. Symptoms that we see with influenza, some of it we may start off think maybe it's a cold, but it's definitely more pronounced and more serious symptoms when we think about this. And there's also some potential for other complications. Rye syndrome with the treatment of littler kids. I believe it's 12. I don't know why that's a magic number, but if you are younger than 12 and you've got fevers and issues, and if it's the flu, definitely concerns, but other things, treatment with aspirin to potentially bring something like this down may set the stage for Rye syndrome. And now ill-advised to use that. There used to, baby aspirins and things like that that were given to little kids. And now we think about other things, Tylenol and Motrin and other stuff that prevents these problems, right? But definitely some concerns. All right. As we move over to measles and our German measles, so rubeola and rubella, we see our MMR, measles, mumps, and rubella as some vaccine culprits. Mumps we will see later in the GI chapter. MERS and SARS, and technically we could have our coronavirus right in here, but these both are coronaviruses with, as we think about, our pandemics, similarities with both of these kind of combined and potential for fatal. Shingles and then viral pneumonia. So bacterial pneumonias, we've already talked about a plethora of them. When it comes to viral pneumonias, a handful of different viruses that may set the stage for these potentials as well. Real quickly, I've only got a minute, but I want to show you just a couple fungal infections. These we think about disseminated situations in this first case, starting in the lungs and then moving to the skin. This is a systemic infection, Coccidioides immitis. There are a handful of other dimorphic fungi that may set the stage for some of these issues. And here we can see some of that. I mentioned the amoeba and the Legionella, and part of me is like, I don't know why this is put right here in the PowerPoints. I think there's maybe a text box that goes through and describes it, but has nothing to do with any of this fungi. There's blastomycosis, blastomyces dermatitidis, a different systemic fungal infection. The mold releases spores. You inhale the spores, which convert into these yeast cells within your lungs, and then it sets the stage to potentially disseminate. So in the respiratory tract, we're thinking pneumonia-like symptoms, but we also could flip it back to the previous chapter and think about skin infections as well. We talked aspergillus. I'm so close to done. Come on in. The aspergillus with the secondary lesions. This is the fungus ball that I mentioned before. So a primary infection, inhaling some of those spores, and then it starts to grow in the lungs. These are a couple of yeasts, pneumocystis in the lungs, and cryptococcus, both of which producing some pretty big capsule layers, and some we think about important with HIV and the progression to AIDS. Farewell.